In this lab, we will investigate the effect of ultraviolet light on bacteria growth. UV light has a shorter wavelength than visible light, therefore it has more energy. When UV light hits the cells, it damages DNA by causing the DNA bases thymine to covalently pair with adjacent thymine bases. This results in mutations and eventually the cells die. The purpose of the lab is to determine the effect of UV light on two different bacteria species. One bacterium produces endospores and the other does not produce endospores. Before we start, I want to explain a little bit about endospores and how they are formed. Endospores are structures produced by some bacteria in response to harsh environmental conditions like drying, radiation, or lack of nutrients. When a bacterium undergoes harsh environmental conditions, it produces a dormant and highly resistant structure to preserve the cell's genetic material during harsh environmental conditions. A bacterium can stay as an endospore for a long time, for example, several decades or centuries. During the time the bacterium exists as an endospore, it is not metabolically active, but when conditions improve, the bacterium germinates and resumes metabolic activity, including reproduction. There are two terms I want to emphasize here. The normal metabolically active bacterium is referred to as the vegetative cell, while the opposite is the endospore. The resting non-active bacteria form is referred to as the endospore. Endospores are produced primarily by bacteria in the genera named Clostridium and Bacillus. The materials you'll need for this lab are an inoculating loop, a Bunsen burner, 4 trypticase soy agar plates per group, 4 index cards, a UV hood, a timer, and laminar flow hood with UV light. You will also need these bacteria cultures. 1 SE slant. This species will represent vegetative bacteria because it does not form endospores. We'll also need 1 BC slant. These bacteria species form endospores. In this lab, we are comparing the effect of UV light on endospores and vegetative bacteria. The hypothesis is that the endospores are more resistant to UV light compared to vegetative bacteria. To test our hypothesis, we will find the minimum lethal dose for these two bacteria. The minimum lethal dose is equal to the minimum amount of time necessary to kill the bacteria. Every student will get 4 trypticase soy agar plates. 2 of these plates will be used for finding the minimum lethal dose for the endospore-forming bacteria, which in this case is Bacillus cereus. On the bottom of these 2 plates, students will write the label BC. This is an abbreviation of the names of the bacteria Bacillus cereus. The other 2 plates will be used for finding the minimum lethal dose for the vegetative bacteria, which in this case is Staphylococcus epidermidis. On the bottom of these 2 plates, students will write the label SE. This is an abbreviation of the names of the bacteria Staphylococcus epidermidis. Using a marker pen, students will draw a line down the middle of each plate. By doing this, the plates are divided into halves. Start with the SE set of plates. Students should label each half of the plate 0 minute, 1 minute, 5 minute, and 10 minute. This represents the length of time the bacteria on the plates will be exposed to UV light. The next step involves swabbing the plate. Using aseptic techniques, swab the plates with SE and BC bacteria respectively. The aim is to cover the whole plate with bacteria. Get a loop full of bacteria from a bacteria slant and run it down the plate from top to bottom. Then, perform a zigzag streak from left to right without flaming the loop in between the streaks. The key to swabbing the plates is to swab the whole plate evenly so as to create a bacteria lawn. Put the plates under the UV hood and expose them for the appropriate time. Please observe safety precautions while working with UV light. Do not look directly at UV light sources. Use index cards to shield the part of the plate you do not want exposed to UV light. During exposure to UV light, the plates should be opened by removing the lids. Remember, UV light does not penetrate. If you leave the lids on, the UV light will not penetrate them. For example, for the plate labeled 0 minute and 1 minute, we will cover the half labeled 0 minute and leave the 1 minute side exposed to the UV light. For the plate labeled 5 minutes and 10 minutes, we will put the plate in the UV cabinet and expose it for 5 minutes. After 5 minutes, we will cover the 5-minute half with an index card and leave the 10-minute part to be exposed for another 5 minutes. Remove the plates from the UV hood and incubate them in the incubator at 37 degrees Celsius overnight. Remember, we always incubate with the plate agar side up. We can see that it took a very short time for UV light to kill the bacterium Staphylococcus epidermidis. This is because this species does not form endospores. This species is its vegetative form. From this experiment, we can see that the bacterium Bacillus cereus has a long minimum lethal dose compared to the bacterium Staphylococcus epidermidis.